Okay, welcome everyone. I'm Dan Friedman. I'm the program manager of Let's Learn. And as most of you know, Let's Learn is a global learning community which works on the premise that everyone has something to teach and everyone has something to learn. And we offer mostly weekly, sometimes more often, a free classes, workshops, conversations, performances that cross political borders and cross cultural borders and work on the assumption that we all can learn from each other. And we have a great workshop lined up for you right now. So I want to, and of course, it's all free. I forgot to say that. That's one of the most important things about it. It's a co-sponsored or it's being co-developed by the Eastside Institute, which is based in New York, but which is really at this point a very international network that does trainings and research into innovative ways to bring about social and cultural change. And the University of North Carolina at Greensboro's Lloyd International Honors College. Together we bring this out to folks all around the world. And let me now introduce to you the Dean of the Lloyd International Honors College, Dr. Omar Ali, who will be introducing out today's teacher.
Thank you, Dan. I'm wearing a tie because I'm in costume as a dean on a Saturday midday. Thank you so much for that. It's great to see everybody here. Welcome. And I have the pleasure of introducing our presenter, who's going to be speaking about the beautifully complicated science of turning plants into medicines, Dr. Nadja Cech, who is an analytical chemist who leads National Institutes of Health funded natural products medicinal chemistry lab as distinguished Sullivan Professor of Chemistry at the University of North Carolina here at Greensboro. Dr. Cech serves on the Honors Council, the Curriculum Advisory Board of Lloyd International Honors College, and has collaborated with me, among many other people, on a range of projects. We've actually taught a course together over a number of years called How Do We Know What We Know? Power Epistemology and Methodology. And we've done a number of things together, but she is just a wonderful partner, and she's partnering with us today by doing this presentation. So with that, let's welcome Dr. Cech.
Hi, everyone. Thank you so much for the introduction, Omar, and thank you everybody for spending a little bit of your Saturday with me. I feel honored by your presence, and I'm excited to tell you a couple of stories and also have some conversation with you at the end. So I have some slides to share. I have a couple of videos to share, so you can sit back and listen. If you have questions that come up as I talk and you want to use the raise hand function, you can. But also you can just write things down or think about them, and then we can have conversation afterwards. So I'm going to go ahead and start by sharing some slides. So hopefully you can all see my slides. Excellent. Okay.
Your screen.
So here we have just my title here, as Omar mentioned. Thank you for the very kind introduction, Omar. And before we jump into talking about some of the research that I've been involved in and some of these questions I want to address today about how we turn plants into medicines, I want to acknowledge the people who are the ones that actually provide the labor that does the great science that I then become associated with. So this is a photo of some of the research students in my research lab taken a couple of years ago. There's a constant influx of people coming in and moving on to other places that are part of the research that we do with plants and plant medicines. And so I get the fun of talking about it, but they do the real effort of doing it. So the credit really goes to them, both for what they do and how they do it and the ideas and creativity they bring to the process. So I wanted to start actually by telling a story about a tree. And this tree, don't know if any of you recognize it, but it is called Taxus brevifolia, otherwise known as the Pacific yew tree. And the Pacific yew tree grows in the Pacific Northwest. I actually knew this tree very well growing up as a child in the Pacific Northwest. And it has been used for undocumented by indigenous peoples of the Pacific Northwest, but it's also famous more recently because it was used to isolate this molecule right here. And this molecule is called Taxol. And Taxol may or may not be a word that you're familiar with, but if you know someone who has been treated for breast cancer or ovarian cancer, they have been treated by this molecule. So the molecule actually became a pharmaceutical drug in the early 1990s. And prior to that, the prognosis for someone who had ovarian cancer was that they were likely not to survive more than a year. About 15% of patients diagnosed with ovarian cancer would survive for more than a year. And now, because of Taxol and other therapies that it's used in combination with, it's more like 85% of people diagnosed with ovarian cancer survive for a year. So it really changed the landscape for oncology when it comes to treating diseases that are actually particularly impacting women. And the discovery of this drug has saved thousands and thousands of lives. And so I wanted to talk a little bit about how this drug was discovered as an entree into this idea of making medicines out of plants. So this right here is Dr. Mansukh Wani, and he was the chemist who worked at the Research Triangle Institute, which is actually not far away from where I'm in. I'm in Greensboro, North Carolina, and the Research Triangle Institute is about an hour away near Chapel Hill, North Carolina. And he worked there with his colleague, Dr. Monroe Wall. And he was part of an initiative by the National Cancer Institute that began in the late 1950s, really began in earnest in the early 1960s. And they screened 114,000 samples from plants collected all around North America, testing them to see if they could kill cancer cells. And one of the plants that was discovered as part of this screening effort was the Pacific yew tree that was collected in a park in Washington State by a botanist. And Dr. Wani was assigned the job of trying to find the molecule in this whole complex plant extract that was responsible for killing cancer cells. And in this quote here, this is a quote from Dr. Wani, he says that it took him 1.5 years to isolate 0.5 gram of Taxol. So to give you some perspective on this, he had 30 pounds of Pacific yew bark that he was working with to try to isolate. And all he got out was 0.5 gram, which was probably about enough Taxol for you to see, like maybe in a bit of like less than a teaspoon of salt in the palm of your hand. That was about 0.004% yield. So about 0.004% of the original material was able to be isolated as this molecule. But beyond that, he didn't just have to isolate it, he had to figure out what the structure was. And those of you who aren't chemists probably look at this thing and say, I don't know what that means. It's a molecule. Even as a chemist, sometimes I'm not exactly sure what it means, to be honest, but all those are atoms connected together in various ways. And Dr. Wani's job was to figure out from that pure material that took them 1.5 years to get, how are all those atoms connected together? Which atoms are there? And what's their 3D orientation in space? And he had to do all of that before anyone would consider looking at Taxol to develop it into a drug. And it actually took Dr. Wani 5 years to go from once they even had that pure material, which remember took them 1.5 years to isolate. It took him 5 years to figure out the structure of this molecule. Now, he was doing that in the 1960s when the technology for doing that was not nearly as powerful as it is today. But to me, it's pretty amazing that he was even able to continue the momentum to work on it for that time period. And it's particularly amazing because the National Cancer Institute that was funding his research really wanted to see quicker progress than that. And in fact, his boss told him, stop working on Taxol. We're not getting good results from this. You've got other things to work on, put it aside. And he said, no, I want to keep working on it on what he called a low priority basis. And he kept working along on it on weekends and evenings, and eventually figured out the structure of this molecule. And then based on that, biologists studied what it did to cancer cells, found out that it had a new mechanism of killing cancer cells. And then eventually, through the efforts of many scientists really around the world, it was developed into a drug in the 1990s. So that's the story there. I want to actually just play for you a little clip of a video from Dr. Wani. Oh, and I see that Nick has his hand up. So before I do that, Nick, did you want to ask a question?
Well, so this can be, you can take this now or later, but I often wonder sometimes when you hear some of these discovery stories, what's the difference between being, inspired and pushing through and being crazy and a little bit of a fanatic, right? You're going to spend your weekends and nights working on this, and people are like, yeah, he's like wasting his time, and
Yeah.
And so, and maybe we can discover this as the talk goes along.
Yeah, what an excellent point. I mean, maybe in some ways the difference between being crazy and a genius is whether or not it amounts to anything. But a lot of times you don't know if it's going to amount to anything. So I guess at least in my experience of being a scientist, part of being a scientist is continuing to pursue your curiosity and staying the course on a question, even when people tell you to stop. So yeah, really interesting point. I love that. All right, I wanted to share just a little bit from Dr. Wani's words, and I'm going to do that now. So bear with me as I switch to video here. Let's see. I'm going to share this. Let's see. Can you all see that?
Yes.
Yes.
All right. Let's see if I can play it. This is just a little clip from a video taken about 7 years ago of Dr. Wani talking about discovery of Taxol.
58. In 1960, they decided to evaluate the potential of natural products in the discovery of anti-cancer agents. This was primarily because there was a lot of public pressure on NCI that they should evaluate the natural products for the discovery of anti-cancer compounds, and they would always cite examples of countries like India, China, Latin America, Africa, where for centuries herbal products have been used as medicine. The officers at the National Cancer Institute were not too excited about exploring the potential of natural products for anti-cancer agents because maybe they were prejudiced and they said, yeah, nothing is going out of it. It will be just waste of money. But because of the public pressure, they decided to start the evaluation of natural products as potential anti-cancer agents, but on a very limited basis with limited funding. They didn't want to spend a lot of money. And therefore, initially they decided to evaluate the natural products readily available, that means from US flora. They didn't want to go to China or India for bringing one plant material from there. That would have been too expensive. That is how NCI decided to support this natural products program.
So I share that partly because I think it's nice to hear the voice of Dr. Wani, who actually did this work. Dr. Wani actually passed away about 3 years ago, but was really a great mentor to young scientists and a good friend to me later on in my career as I was studying natural products research. And I'm really inspired by his story. I think it's really interesting that he mentions this prejudice in that clip, which was part of the reason I wanted to share it. Because as someone who studies plants, as medicine, even now, after such discoveries have been made like Taxol, that clearly have saved thousands and thousands of lives, there's still kind of an attitude that maybe this isn't something worth studying. And so that's something that I think about. And really, I think partly that attitude dates back to the late 1800s, at least here in the United States, which was a time period when there was a schism in how medical practice was done. And there was a medical practice when it was imported to the US from Europe. The idea, Western medical practice, the idea was to use practices that were common in Europe, such as bloodletting and leeches, high doses of mercury, which we know are not a very good idea, and high doses of opium. And there were also a group of doctors in North America who had this idea that it would be better to rely on other ways of curing disease because these methods weren't necessarily very effective. And they turned to indigenous practices here in North America. And there was a real focus on this idea that it would be North American plants that they would use. I think that partly aligns with sort of the manifest destiny thinking that was going on at the time, all kinds of interesting alignments of history there. But what ended up happening was that a whole new school of medicine was developed called the eclectic school of medicine in North America. And the eclectic doctors relied on indigenous knowledge, spoke to indigenous healers in North America. Of course, there was indigenous healers all around the world, but they were focused in North America. And they developed a whole pharmacy effectively based on plants, and they would prescribe plants to patients for illnesses. And they ended up actually having schools of medicine that were very active in the late 1800s and the early 1900s. But what ended up happening then around 1920 is that penicillin was discovered. So you all know that was sort of a revolution in medicine, that you could use this single molecule. So this is before Taxol. This was really the first time that you found this single molecule that could be used as a medicine. And people got really excited about the idea of isolating molecules from their natural sources and using them as drugs. And so they sort of, the medical association wanted to distance itself from the eclectic doctors. And in fact, the American Medical Association forbid their members from consulting with eclectic doctors. And eventually, the eclectic school of medicine that was using plants was phased out. And what we see today in Western medicine is a focus on either synthetic compounds or molecules that originally come from natural sources, like fungi and bacteria and plants, in their isolated forms being used. And there's sort of an attitude that, oh, that plant stuff, that's kind of weird hippie stuff. I don't know, herbal drugs, you don't really know what's in them. But that line that's drawn is actually not nearly such a concrete line, because a lot of the practices in Western medicine are based on indigenous knowledge. So I come into this story, and I'm going to share my slides again and with a few photos. Let's see. Here we go. Okay, hopefully you all see my slides again. So I come into this story right here, and I actually can't see all of you. So can someone just tell me if you can see the slides?
We can see it.
Okay,
Yes.
thank you. All right. So I come into this story. This is me in 1983 with my family living off-grid in Oregon. My parents, if you can't tell from the photo, were hippies that decided they wanted to raise us off-grid. And so I came into an understanding about plant medicines from a totally different perspective, which was the perspective that was informed more by studying what eclectic doctors had done and what indigenous people had done prior to that. And
Which one of those children is you?
this is me right there. I don't know
Okay.
if you can tell. In a dress that my mom made for me. And so I grew up around plants and grew up loving plants. This is actually me sitting next to my brother in front of my first garden that I grew. And you can see I've decorated my hat with flowers. And I loved this idea of plants and humans interacting together, and over time began to become interested in the science of what was going on when we consume plants or use plants. And it turned out that I had the opportunity to, this is me again now in 1992, attending Rogue Community College. So up here at the top says Rogue. That's me and my little sister with our matching fanny packs. And I got the opportunity to go to community college and start to study science. And I met some wonderful scientists who encouraged me to study science, and eventually ended up going to graduate school at the University of New Mexico to work with this guy here, Chris Enke. And Chris Enke invented a type of mass spectrometer. And to put it simply, what mass spectrometers do is they are able to weigh molecules, which gives us information about the structure of molecules. So remember, I was telling you that one of the challenges with Dr. Wani's work was that the instrumentation wasn't very developed in how to figure out the structure of molecules when he was studying Taxol. And so it took him 5 years to figure out the structure of Taxol. But the types of inventions that came along this was in the 1970s, this type of mass spectrometer was invented, really made it easier to assign structure to molecules. And I was fortunate in graduate school to get to work with these types of instruments. Actually, this is me with Dr. Enke in 1997. So I've gone from community college to graduating with my PhD from the University of New Mexico, working with Dr. Enke. And then in 2001, I came to the University of North Carolina, Greensboro, which is where I am now. And this is me sitting in front of my very own mass spectrometer. So I've used my funds they gave me when I came to UNC Greensboro to buy a mass spectrometer. And I would describe myself in this picture as a hippie with a mass spectrometer. So what is a hippie with a mass spectrometer going to do? What I wanted to do was study plants and figure out how the molecules from plants could be used to treat disease. It seemed like a natural question to ask given the history that I had had. And I had to pick a plant to work with when I started. And the plant that I chose is actually shown in the back of this pickup truck. It was a plant that I had a lot of experience with. So this is me again. Now we're going back in time just a little bit to the year that I transferred to university. And we sold the plant that's in the back of this pickup truck, this whole crop, which I had actually grown from little seedlings myself with my siblings, and planted them out in the field, taking care of them for 2.5 years. Sold the roots for $5,000, which was enough to pay for my going to university. And my parents used the entire proceeds from that crop to pay for me to go to university. And the plant in that picture is Echinacea purpurea. And some of you have probably heard of Echinacea before because it's very commonly used. In fact, it's one of the most high-selling dietary supplements in the United States. And speaking of, I was talking about the eclectic doctors earlier who had these schools of medicines based on plants. Echinacea is a plant that grows in the prairies and plains of North America and was very prominent in eclectic medicine. It was one of the main medicines that was used in that time period. And it was also sold, it continues to be sold in various forms. You can buy Echinacea pills, but you can also buy something that looks like this, which is an Echinacea tincture. And I grew up not only growing Echinacea to be sold, to be used to make medicines like this, but also using Echinacea myself. And part of the reason for that was that my dad, who's shown in this picture, Nadja Cech, who is a very well-known herbalist and grower of medicinal plants, was really interested in Echinacea, and he was also interested in the research around this plant. So early on, he started to talk to me about the eclectic writings about Echinacea. And he also told us, I don't know, I wish you were all here with me because I would share some Echinacea tincture with you and have you taste it. But if anyone has ever consumed Echinacea, what it does is it makes your whole mouth tingle and go numb. It feels like you have a mouthful of buzzing bees when you consume Echinacea. And I remember very distinctly consuming Echinacea as a kid and having my dad tell me that tingle, the tingling sensation was due to this molecule called alkylamides. And at the time, I didn't know what an alkylamide was. I would never have been able to draw the structure that you see here, but I learned the word, I memorized it, and I was like, okay, alkylamides are important. So many years later, when I had my own mass spectrometer and I was going to study plants, I thought, well, let's study Echinacea and let's study these tingle molecules in Echinacea. Part of the reason I wanted to do that was because my dad told me that the tinglier the Echinacea was, the better the medicine. And so I believed that, but I wanted to understand the mechanism for how that would really happen. And my thinking was that we would add some validity around this concept of how a plant can be used as a medicine by studying the molecular the molecules present and figuring out what they do, much as the studies were done with Taxol, with Pacific U, finding Taxol and then developing that into a drug. So the question that I started out with in my research, and research, scientific research always has to start out with a question. And the question I started out with was which chemical constituents or which molecules in echinacea are responsible for its effectiveness in what it's used for, which is treating or preventing infections. And so what are those tingle molecules that are responsible for its activity? And what other molecules are there that might be important? So I thought the alkylamides would be important because they were what made echinacea tingle, but we were really interested in anything in that plant that might be important and figuring out how it worked. And I was actually fortunate to get funding. So echinacea has helped me out a couple times. It helped pay for me to go to college. And then also my first research grant from the National Institutes of Health was to study this plant. So we actually got funding to do that back in 2003. And we had this hypothesis that these alkylamides from echinacea stimulate the immune system and prevent infection. So taking a step back here, this a lot of science talk here, but I know you all are capable of following it. I hope you are. Actually, let me stop for a second. I'm gonna stop here. It's so strange because I can't see you when I'm showing my slides. So let me just check in with everybody and see where you're at so far with the story. Are you following me or do you have any questions? It's okay. It's going okay. All right, good. It's so strange to talk to a screen when I don't see people. So I'm just making sure. All right. I'm going to go back to talking to the screen where I don't see people. All right.
It's great. Wonderful storytelling.
You're okay. All right. Thank you. Thank you, Omar.
Nadja,
Okay.
can I?
Yeah, go ahead, Nick. Please go ahead.
So I'm curious, having been raised in a nice suburb of Albany in a development, with my father being a civil engineer, right? I'm curious about your parents choosing to go off the grid. And one of the things I was struck by was the picture of the particular picture of your father that you had. He's got a notebook, right? He is not just like wandering off into the woods, la-di-da, Tom Bombadil, like, and to raise $5,000 worth of echinacea. I don't know how many acres he's talking about, but what was their? Do you have a sense of what their prep was? Like, what was their background? What did
great
how was that
question.
decision made?
So my parents both came from middle-class families. My dad's dad was a doctor. My mom's dad was a tax accountant. They both left city life because it was the 1960s and they wanted something different. And they wanted to get out of this, capitalist way of being in the world. But as it turned out, after spending the first decade or so trying to raise 3 children off grid, what they realized was that we were completely broke, which is the reality of trying to be a hippie sometimes. That is my experience, the reality. So that's when my dad was like, okay, I got to figure out a way to support my family. And so he was also always interested in plants and he'd been sort of studying plants as a hobby. And so he began to collect seeds from plants and sell them and got connected with this company that sold medicinal plant extracts. So that photo I showed you of echinacea was actually echinacea that is sold by Herb Farm Incorporated, which was in Williams, Oregon, the same town where I grew up. So that was kind of serendipitous. And he ended up starting to work there and then getting interested in the writings of the eclectics. So you see him with his notebook, he's recording stories about how plants are used for medicine. That was when he was traveling in Madagascar when that photo was taken. So my dad is like, he's actually did not graduate from college, but he's a very interesting in that he really is a scientist and a self-trained botanist and an herbalist. And I learned a lot from being his child, and I'm sure that's why I am where I am today. Thank you for that question. All right, let's go ahead and I'm going to share just a little bit now about the scientific discoveries we made when we decided to start studying echinacea. So how do I do that? I'm going to do it by sharing screen. share screen, and let's go with this. All right, so we have this hypothesis, which is that alkylamides from echinacea stimulate the immune system and prevent infection. And how do we go about studying that? Well, the first thing that we did was we got some plant material of echinacea and we brought it into my research lab. This is one of my first students that I worked with at the University of North Carolina, Greensboro. This is Brandy with a bunch of echinacea that came in the mail from, you guessed it, my dad shipped it to us. And we collected it and we extracted it in the lab. So essentially like making a tea out of it. Although I think what we did initially was to make ethanolic extract. So you mix it with alcohol and extract the material. And then we needed to figure out what it did. So we analyzed it with our mass spectrometer to figure out what molecules were there. And we wanted to also figure out what those molecules did. And the hypothesis was that echinacea stimulates the immune system. So you all know that your immune system is important when you get an infection for warding off that infection. And we grew immune cells in petri dishes. So this is a photo of macrophage cells being grown in petri dishes. And we added echinacea extracts to them and we let them sit with the immune cells. And we tried to look at what happened 24 hours later. And the response that we measure is essentially when immune cells get excited, they start releasing molecules that are called cytokines. The term isn't super important, just realize that the immune cells over here are sitting here in an unexcited state, and here they are in a stimulated state where they start producing molecular signatures that we can pick up. And then from that, we wanted to compare different echinacea extracts, look at different molecules from echinacea, and try to narrow down which molecules are causing that effect. But it turns out this was much harder than we expected. So I'll give you an example of some of the really perplexing data that we got in one of our early studies with echinacea. We collected echinacea from all over the state of North Carolina. So this is a map of North Carolina showing the counties of North Carolina all outlined in this map. And this is a photo of some echinacea being harvested in a field in Western North Carolina in the mountains. There was also echinacea being grown near the coast and in the foothills and in different parts of the state, all under different conditions. And each one of these numbers represents a site where we collected echinacea. And what we found from the samples was that some samples, when we added them to the immune cells, they inhibited the production of those little molecules that are indicative of the immune cells responding. So they suppress the immune cells instead of inhibiting them. But other extracts from other places stimulated the immune cells. So here we are, same plant, harvested in different parts of North Carolina, having totally opposite effects on immune cells. We didn't expect that. It was really surprising. So what I thought immediately, my chemist hypothesis was, wow, these must have different amounts of those molecules, those tingle molecules that I know about being important. These plants must have different amounts of those molecules in them. But when we went into the research lab and we used our mass spectrometer to study the presence of those molecules in there, and you don't have to understand exactly what's being shown in this picture, but the main take-home message is that the amounts of those molecules in different samples from totally different parts of the state were almost exactly the same. So here I was expecting that the molecules were going to be responsible for the different effects, but they didn't match at all. It didn't map at all. So my hypothesis, our hypothesis that my students and I had put forth was a failure. It did not work. Right around that time, there was a discovery that was super interesting to us. This was published in 2008, a discovery that had to do with the importance of microbes living inside plants. So all of my interest in echinacea up until here had been focused on molecules that echinacea makes. But it turns out that just like people, plants have a microbiome. And inside of a plant are many fungi and bacteria living perfectly happily and in symbiosis with the plant. Just like inside of you, you have bacteria that are helping you digest your food. They're helping protect you against disease. They're potentially affecting your emotional health. There's all kinds of importance of our microbiome. Same is true of plants. So if this is actually a photograph taken from one of the projects that I was doing with a student in my lab, where we took a root from echinacea, and that's what you see in the middle here is a little piece of echinacea root, and we put it on a dish of agar. And agar is just a material that bacteria and fungi grow really well on. And you can see this white stuff all around. That is bacteria growing out of the root. So this is just a visual demonstration of the fact that microbes are living inside of roots. And it turned out that there was a new hypothesis about why echinacea was stimulating immune cells, which is that instead of the molecules from the plant stimulating the immune cells, it might be the bacteria that are doing it. And in fact, this makes a lot of sense because your immune system is actually primed to respond to infection, right? And one of the main things that causes infection is bacteria. So this was a whole new way of thinking about this. And we actually did a few more experiments. I'll just show you a couple more slides of our experiments. There's not much more here, where we actually grew echinacea plants in sterile environments to test this hypothesis. We grew them. These are sad little echinacea plants growing without any bacteria present, without their microbiomes. They didn't like it very much, but we could grow them that way. And what we saw was that for the ones that did not have bacteria, we did not see stimulation of immune cells anymore. So we really realized that everything we thought was happening due to the plant in our immune cell experiments was happening due to microbes that were living within the plant. And this is just a final piece of data showing that. So this is a comparison of a bunch of different plants here on the x-axis. All these are different plants, individual echinacea plants, and measuring how much they stimulated immune cells. And then down here is measuring how much bacterial components are in those plants. And you can see how these match up perfectly, that the higher the ability to stimulate the immune cells matches with the higher amount of those bacterial components. So I'm done showing all these data slides. Thank you for bearing with me through that. But in the end, what I wanted to wrap back around to was this question was, which chemical constituents or which molecules in echinacea are responsible for its effectiveness in treating and preventing infections? There was a lot of assumptions there that I didn't realize we were making when we asked that question. And one of the assumptions was that the molecules were coming from the plant, and turned out they were coming from bacteria. And the other assumption we were making was that echinacea works to treat and prevent infections. And what's interesting is right around the same time that we started making these findings, some clinical studies came out where they did double-blind placebo-controlled clinical studies of echinacea, and they found no effect of echinacea for treating and preventing infections, which was a huge surprise to me. So I guess I would like to say that I came to the end of this research project with more questions than answers. And that's often the case when one is involved in scientific research. And so what I wanted to do now actually is completely switch gears and just share with you a couple little videos about another aspect of how echinacea might be important in human health. So that will take just 1 second here, and then we can open up for a conversation. So here we go. Get ready. Share screen.
Hello, Let's Learn folks. I wanted to use the last bit of this conversation to take you to a place that's one of my favorite places. This is the Dunleith Community Garden. It's a garden that I volunteer at in the Dunleith neighborhood, which is an urban part of Greensboro, North Carolina. You can see there are cars and houses all around here. The garden itself is a piece of land that's been slated for development for the past 30 years, and although the developers continue to say they're going to develop it, they're letting us garden here until they do. And we have about 30 families that grow food here, both for each other and for the neighborhood. It's also a place for a community to gather. These are fig trees. They're dormant right now, but they're covered in figs in the summertime. All the neighbors come and pick figs. There's a swing over here where kids can play. People can sit and meditate under the trees on the swing, and herb plot where people can pick herbs. And I also grow some plants here in my plot that are used in my research. So this is a plant that, rosemary plant that we actually harvested and studied antimicrobial properties of rosemary and published a paper with one of my wonderful students on this plant. And so I thought the Dunleith Garden would be an appropriate place to talk a little bit about the end of the echinacea story. So hold on, I'm going to flip to another video so I can switch this around. Hello, Let's Learn folks.
All right,
I
hold
wanted
on
to
one
use
second.
the last.
All right, escape from that. All right, hold on. One more video. I'm really had to switch to a different mode here. All right, there we go. So, okay, hold on. One more video. This one's short. All right. Here we go. And play.
In some ways, and earlier on in my career, I felt like the echinacea project was a bit of a failure because I set out to figure everything out and understand which molecules are important in the activity of echinacea and unravel all this complexity and get to the right answers. And I think what ended up in this particular research project was a lot more questions than answers. Clinical studies of echinacea still have not shown efficacy for reducing symptoms or severity of colds and flus. And a lot of people are uncertain of whether it actually does the things that people have thought that it did to the immune system, and whether maybe all of that could be an artifact of the way we did our experiments, growing cells in petri dishes and adding extracts to them is just not a very good way of representing what goes on in the whole complex human body. But more recently, I've decided to embrace the uncertainty that comes with my research on echinacea, or the collective research on echinacea that my group and my collaborators have done. And in a way, I've come to find it to be rather comforting to accept that sometimes we don't know the answers, that scientific inquiry is a wonderful pursuit, and it's great to go after our curiosity and find out new things. And sometimes, in the case of the research like what Dr. Wani did, come up with very concrete answers that can be very helpful for modern therapeutic practices. And on the other hand, I think also that it's humbling to realize that whatever answers we do come to are only just a tiny slice of the big, beautiful whole of what it means to be a human interacting with plants and other living things on this planet. And so I've come to be okay with not having all of the answers, and in fact, to really be at peace and to actually celebrate that, like beyond being at peace, to celebrate not having all the answers, to celebrate not knowing. And I would also add that I've sort of expanded my viewpoint of what it means to make a medicine out of a plant. So for a long time, I was really focused on finding molecules in plants that would be useful to put in a bottle and sell to someone that could cure their disease. And these days, I really like to spend a lot of time in places like this beautiful garden with plants that are very healing, whether we consume them or just grow them or interact with them or get to learn from watching them grow and breathe the oxygen that they produce and share our carbon dioxide with them. And so in that way, the question of whether or not echinacea or any other plant that's used for medicine is healing is already sort of a solved question. And the answer to that question is yes. So I'll end there, and I look forward to having some great conversation with all of you.
All right, we're done.
Yay!
So that's the stories. What do y'all think?
Oh, wonderful. Yeah. So the floor is open. Let me just ask you all, if you would, if you thoroughly enjoyed this storytelling / teaching, I want you to perform a very thoughtful beard rubbing agreement. How, like so. Yeah, if you really have enjoyed this, yes, very good, very good. All right, we have a couple of hands up there, I see. So let's start with Lois Holtzman. Dr. Holtzman, what is your question? And then we'll go to Paige.
Thank you. Thanks, Nadja. That was so delightfully informative.
Thank you, Lois.
And the embracing of uncertainty is something I'm very happy with.
That was really my shout out to you, Lois.
Yes. So it seems like what has been left out of all of this study, and that's not a criticism, but is the uniqueness of every person. So we do know that some people are very responsive to all kinds of things that other people are just not responsive to. And we do know that we don't know like what else is going on within the body and all the bacteria around a body. So is anybody kind of looking at that at the totality of the environment?
Such a great point, Lois. I really thank you for asking that. So yes, people are looking at that.
You saw
And
it froze, or everybody froze.
oh, I think we're still good. We're here. We're not
Okay,
frozen.
Okay,
We're here.
I did not.
It was just a
hear you.
momentary freeze.
Okay,
So, a lot of what I've done in other research projects that I didn't talk about today is to think about how it's different when you use a mixture of many molecules versus a single molecule. So in some levels, we can go in increasing complexity from one molecule to many molecules. Then we can think about how a plant interacts with its environment. The bacteria that are there in the plant depend on where the plant was grown, right? And so it actually picks up different bacteria from the environment if you grow it on the coast of North Carolina versus in the mountains of North Carolina, which is why we saw those crazy different results for our plant extracts from different places. And then the plant is interacting with the environment beyond that, which includes us, the people. And so, and of course, we come with different bacteria living inside of us with different metabolic processes. So it's not terribly surprising that you do a clinical study of one extract of one plant with sort of a group of people and try to look at the results, and maybe it doesn't work the way you expect it to. And I think that context is so important. And when we try to average out people to do a study, we lose that context. And sometimes we lose things where somebody's seeing a really important effect and other people are not. So I guess what I think there are people doing that. There are people studying how difference in your genetics or difference in your microbiome affects how you respond to a treatment with a medicine. And that's a very interesting area of research. Sometimes people refer to it as epigenetics. I personally think that developing relationships with plants is really an important thing. And it's also a very personal thing. And if we're growing our food in a garden and then consuming that food, we're consuming microbes that are part of our environment that's different than if we're consuming food that came from a different place. And I have a suspicion that that has an important impact for health, although it's not something I've studied. So yeah, I don't know if that starts to address your question, but I think it's all contextual is what it comes down to. And when we try to do reductionist science, we remove the context, and that does not represent life very well.
Right. Thank you. Thank you. Agree with that. So I read all these new discoveries, or this is good for you, this is bad for you, like just with a grain of a healthy handful of salt.
With a healthy handful of salt.
Right.
And yet sometimes also, I have to go back to, it's pretty awesome that you can get a vaccine and keep you from getting an infection, or you can take Taxol if you have breast cancer and it cures your breast cancer. So it's like, it's all part of the mix, right?
Yeah.
Thank you. Let's go to Paige.
I have so many questions.
You, Paige, hi.
Hi. This is just stirring up so much biology for me, especially with the symbiosis of the bacteria and the plant. Did you do species composition for the microbiome? Were you able to do a comparison between coastal and mountain echinacea?
We didn't do it as part of that study I showed you, but we did a different study where we collected echinacea from different environments and looked at the plant microbiome. And what it comes down to, Paige, as a great biologist, you are insightfully picking up on, is that microbiome is totally reflective of the environment. So I actually naively thought when I started studying microbiome in plants that it was going to be the plant species that was going to dictate the microbiome. I was like, oh, certain species are going to have other species living with them, and we'll be able to figure out which species are important for the activity of echinacea. But it turns out that what matters way more than what plant species you're looking at is what environment. So, for example, if you dug up an echinacea root and then you had a carrot growing next to it and dug up that carrot root and you compared the microbiomes, they would probably be way more similar than if you dug up echinacea in one place and then dug it up in another place and tried to compare the microbiome. So yes, we did species analysis on echinacea as part of this research, and there's great diversity in what we're able to pick up. And we were just looking at culturable bacteria. If you actually looked at this based on techniques that allow you to sequence what you can't culture, we would have seen even more diversity, I'm sure. But it really comes down to environment.
So does your hypothesis take you to think that it's the bacteria themselves that are triggering an immune response, or is it a byproduct of their reactions within the plant?
I think it's the bacteria in the experiments we were doing. I think it's the bacteria themselves. There's a component of the bacterial cell wall, LPS, that's well known to stimulate an immune response in bacteria. So I think we're adding bacterial cell walls. The bacteria don't even have to be alive. You add them, there's components of their cell wall that stimulates bacteria. But whether that's relevant in the human body is a really different question, because you're not necessarily getting those bacterial components into your bloodstream. And then there's this whole other part that I didn't get into too much, which is the immunologists think that it's maybe better to suppress the immune response than stimulate it when you have an infection. And so this whole idea, we kind of got sent off on a red herring, I think, as scientists studying echinacea, because people saw that when they took immune cells and added echinacea, immune cells were being stimulated, and they were like, yay, stimulate the immune system, prevent infection. That's not really what you necessarily want to do. So it's, I don't want to take too much time, but Paige, you and I could talk more together about this stuff for probably hours. It's super nuanced and interesting.
Thank you.
Thank you for your great questions.
Thanks, Paige. Let's go to Karen.
Yeah, can you hear me?
Yes, we can.
Yeah, I was wondering, since plants have been used by indigenous populations around the world, you started out with that. Starting from that, did Dr. Wani select that particular tree because of being pointed in that direction? I mean, there's billions of plants. How do you select the one unless you have some indication from something that they've been effective? I mean, you just kind of picked echinacea or echinacea because it's been known in the literature that somehow it improves response to, or is a preventative kind of thing. But we don't yet a lot of the research has come of all of these things have been faulty. A year later they find out that it isn't effective at all. But I'm interested in how you select. Do you use what is known from the indigenous populations that have used those substances for years?
Karen, that's such a wonderful question. I'm glad you asked it. So the National Cancer Institute project, you remember I mentioned that they found Taxol from screening 114,000 plant extracts that were collected in North America over a period of about 20 years. And when they set out to do those collections, they did look at the traditional, what we call traditional, I never know exactly what to call it. But ethnobotanical is one word people use. The knowledge, indigenous knowledge is about how plants have been used. That was one thing they looked at, but they also collected a lot of samples somewhat randomly. Just like botanists just going out around North America and collecting whatever they could and putting it into bags and sending it off to be tested. And so it turns out that Taxol in this was selected not because of its traditional use, but it was used. So, I think that in that case, with the Wani study, the screening was just based on let's get as many things as we can into the screen and see what works. But there's literature that shows that if you choose the plants you screen for whatever you're testing for, like when I say screen, what I mean is testing to see if it kills cancer cells, but you could do other kinds of experiments. You could see what it does to immune cells. You can see what it does to bacteria. The literature shows that you get much higher hit rate, much higher success if you rely on knowledge of how these plants have been used throughout time than if you just start doing it randomly. And of course, it makes sense, right?
That's the reason I asked that. I mean, to think about the billions of plants in the universe and just randomly pick up one, it doesn't make sense. I mean,
I agree
at all.
We should, and really there's this separation. This is why I struggle with the terminology here, is that there's this separation between the indigenous knowledge and the quote scientific knowledge, but all the scientific knowledge is relying on the knowledge of people, and we're all people that have been studying this stuff as long as there have been people, right?
Well, the other thing is the selection of what your target is.
Sure.
Targeting the immune system, oh my gosh.
Could it be more
of course,
complex?
we're all individual immune systems, and targeting a cancer cell is a little bit more specific. Targeting bacterial cells or viral cells seems to be much more specific and maybe would lead to some information.
I cannot agree with you more, Karen. I started out studying echinacea and the immune system, and these days what I study is bacterial cells because
absolutely,
the immune system is too darn complicated for me.
That's right. They're specific.
Yeah.
Thank you. Dan.
Hi, I have probably two very naive questions because I'm not a scientist. One is about Taxol. When you mentioned it killing cancer cells, you talked about uterine cancer and breast cancer. Is there a reason why it doesn't attack all cancer cells?
It is used in several other types of cancer, but it seems to work well against sort of moderately fast replicating cancers. So, and actually some of the initial screening for Taxol was done against leukemia, which is a much faster replicating cancer cell, and it showed no activity. And so people thought maybe it wasn't going to be a good lead, but that's kind of another story. But it turned out that it does very much what you're narrowing in on there, Dan, is that it very much matters what kind of cancer cells you're looking at. And so that's actually not a naive question. It's a great question. And one of the simple things is how fast replicating the cancer is, but there's much more to it. Cancer is obviously not one disease, it's many diseases. And the different therapeutic strategies for different types of cancer depend on the properties of those particular types of cancer.
Yeah. And then my question with the echinacea, I think was already answered. I think this is a more crude version of what Paige was asking you. So since it's the bacteria, would that same bacteria, if it was in, rosemary or wheat, would it still have the same, or is there something in the interaction between the echinacea and the bacteria?
I love that question, Dan. That is really getting to the heart of it. And the answer is, I don't know. I don't even know if bacteria from echinacea are specifically important when we consume echinacea, because I don't know because bacteria from echinacea are different every single time you grow echinacea. So I don't think there's an interaction between the plant and the bacteria that I know of, but we can't rule that out. That's another great question. That's why I say with this project, we were left with so many questions. Are there certain bacteria that are specific to echinacea that we just don't know about that are important? Is it really not the bacteria at all that we want to look at, but the molecules that are suppressing instead of stimulating the immune system? And that would maybe be these tingle molecules I talked to you about earlier. Is it true that it's actually an effect that happens in the digestive system where you consume these plants and it's interacting with your immune cells in your intestines? And that's important. And so it doesn't matter if it gets into your bloodstream. Like there's so many questions on this project. But my feeling about it is that when you eat echinacea, you're not eating necessarily bacteria that are specific to echinacea, but you are consuming bacteria. And that's true every time you consume a plant, every time you eat a salad, every time you eat a carrot, you're consuming bacteria. And I would guess that that's important for human health. Like, where does our microbiome come from, right? So I'm really interested in the relationship between humans and plants and how that relates to agriculture, because we've changed our agricultural practices over time to shift away from growing our own gardens and fertilizing them with manure and things like that, to having fossil fuels growing big fields of wheat and corn. And I'm sure the microbes that we're consuming are very different given that. So there's all kinds of great hypotheses you could come up with there, but I haven't had the opportunity to test them yet. But great questions that were not at all naive, but just awesome questions, Dan.
Thank you.
Let's go to Nick.
Hi, Nadja. Like everybody else, I have lots of things I'd like to get your opinion on. I'll give you a choice.
Okay.
You want the question on reductionism or the question on like mitigating risk in choosing your research project?
Oh, I want both. And don't make me choose between those fascinating questions. I got to take off my jacket for this.
Oh no. Just to roll up your sleeves.
Yep.
Let's do the mitigating risk, because that came up with I was talking to some students about how to manage how to develop a research statement and potentially a proposal, right? So like and I'll ask it this way. So when in the hippie with the mass spec, right, sort of thing, right, you're new faculty member, you've got a graduate student for the first time. Were you thinking about did you perceive that there was a risk in taking this project on, that you would get a null result, or would fail, and you wouldn't get any publications, and your graduate student wouldn't be able to graduate, and and how do you manage that when you're looking at some of these questions?
Great question. I'm glad I didn't know. I'm glad I didn't know what I was getting myself into when I was sitting there in front of my mass spectrometer back in 2001, starting to study echinacea. I'll tell you what I said to my dad when I called him on the phone and asked for some echinacea. I said, I want to study echinacea for a couple months and figure that out, and then I'll move on to something else. So that's how naive I was, Nick. It was like I had no idea. I had spent my entire graduate work studying molecules that you buy out of a bottle and never thinking about biological interactions at all. I was interested in it, but I had no experience with it. All I knew how to do was run a mass spectrometer. So I really had no idea what I was getting into. Thank goodness, because I never would have done it. The immune system, as Karen was pointing out, is about the most complicated thing you could try to study. Yeah. And it was a terrible thing to start with, but I did it anyways because I didn't know any better, and I learned so much. So, and we did publish. I will say that even though our results were not exactly what we expected them to be, we published 15 different papers on echinacea over a period of 10 years. So we still learned a lot of interesting things. And who knows how those impact other research down the road. But it is definitely true that it's important to think about mitigating risk when you're embarking on a project. And I try with my students now. I've been doing this for 20 years now, so it's very different. I'm a very different person than I was when I started. And I really try to pick 2 different projects for my students. One that I think is almost certain to give us some results that we can publish in a short time frame, and one that's a long shot, but if it works, wow, that would be amazing. And that's kind of how we go about it, is we try to do both. And sometimes the thing that you think is going to be easy, though, turns out to be hard. So it's not, there's no guarantees in science, right? But yeah.
I love it. I'm just going to figure this out in a couple of months. No problem.
Just a couple months, and I'll move on to something else. Yeah. That's how naive I was, Nick.
Yeah. I love it. I was just thinking, I've actually never heard you say that you have, in a systematic way, you have the students pick 2 things, one that's doable and the other one that's more aspirational, or sort of like that. That's really interesting. Yeah, so if other folks want to jump in with questions, but I'll ask you one thing, which is that part of, I think, also like the way we do this sort of division of Native American peoples or indigenous, traditional medicine versus scientific medicine. I know that, we've had conversations about that division, which is that, in some ways, people have been asking questions, like the way that hypothesis-driven modern science is organized and funded, right, as you've taught me. But people have been asking all kinds of questions. They just don't write it down and publish it. And so there's kind of I wondered if you could speak a little bit to the blurring of the lines between pre-scientific and quote scientific, research as in relation to natural products medicine. Could you say a little bit about that, the blurring of that?
Yeah, I mean, they're very, very blurry. You said it really well, Omar. I think the best way to figure out if something works is to try it on a human, right? Because when we try to grow, for example, especially like the complicated example I was giving where we're growing immune cells in a petri dish and then trying to translate that to what happens in a person, it's tough to make that translation because there's so much more complicated things that go on in a person. So people have been doing research on people as long as there were people. And, I'm sure a lot of people died in the process. It frightens me to think about, how people figured out that certain plants were poisonous, for example, right?
Right.
So we should not ignore all that research that people gave their lives for and were very careful to do of documenting what happens when you consume particular plants. There are so many examples of drugs that were developed based on knowledge that predates, quotes, the Enlightenment, the scientific revolution, however you want to say it. One of them is the best treatment for malaria, which was developed by a scientist in the 1970s in China, Tu Yu, as artemisinin. And she went back to 2,000-year-old Chinese texts to figure out how Chinese doctors were treating fevers and found this particular plant tea that they were making. And then she studied it, and eventually they isolated this molecule artemisinin that's now saving, again, thousands of lives around the world because it treats malaria. And people were curing malaria before they knew there was such a thing as this molecule artemisinin. So we have this attitude of like, well, if you haven't written it down and put it in a paper and figured out the structure, then it doesn't really count.
Right.
But I mean, I don't know, 2,000 years ago, if I had malaria and I went to a Chinese doctor and he gave me artemisia plant and it cured my malaria, I would have probably called that a success. So I don't think it really makes sense to somehow poo-poo that knowledge, even in comparison to today's knowledge. So yeah, I think it's very blurry, and yeah.
Thank you for that. Yeah. I have another thing I want to ask you about, or actually, I love the way in the second video where you're sitting and talking about how you said that you don't know exactly if echinacea works in a particular way, but that there's a healing process in being in the garden. And I was thinking about like part of what you do as somebody who leads the Czech Research Lab is you're training all kinds of people, and you have been doing this for now two plus decades, that are going to go on and do all kinds of other things. So we think so like, okay, are we going to solve the cure for this specific thing? But
Right.
The impact of what we do, we may not find the cure for this, but what are we also figuring out in the process? So I was just reflecting on I really like the way that you articulated that.
Thank you. Yeah, absolutely. There's sometimes we can get a little stuck on one idea and get closed off to other opportunities.
Yeah. Amber, you're nodding. What were you thinking, just out of curiosity?
Oh, just how it's just a common thing that you hear about with scientists, or with science, I should say, where, everyone's looking at the data with through a certain lens, and then but when it's sort of looked at through from a different understanding, or in a naive way or whatever, that's when sometimes discoveries are made, when you look at it, a different way.
Yeah, so beautiful, Amber. I agree so much. And I think also that we think we feel very attached to the idea of science as the answer to questions in our sort of modern society, but as if science is one thing, but a lot of times I like what you were saying about looking at the data. We can look at the same data and draw very different conclusions depending on our perspectives. And this idea of objectivity, I don't think really is such a thing. So it's great to have many people looking at the data, is I guess one thing I would draw from that, and get multiple perspectives.
Yeah.
And talking about multiple perspectives, about half the class has not asked any questions yet, so I wanted to encourage them to do so.
We've gotten great questions. I have to
Oh yeah,
say.
we've gotten great
Thank you
questions.
all.
I'm not saying they weren't great. I just wanted to just open the door to anyone else who might have a question or a reaction or a thought off of what we've been doing.
Yeah.
Yes, Ruben.
Ruben, hi.
So, hi. So now that we don't have echinacea to strengthen our immune system, what do we got? What's left for us?
Well, I guess I wouldn't go so far as to say we don't have echinacea to strengthen our immune system. What I would say is I wasn't able to show what I expected to show with my work, and others have also not been able to, but I'm still holding out the possibility that echinacea is doing things we don't understand. But I think that's not the question you're asking me. The question you're asking is, what else can you do for your immune system? I mean, I think when we talk about immunity, a lot of the simple stuff is probably the most important. Like, for example, sleeping every night, having daily exercise, having a practice that helps you not be so anxious and stressed, whether that's working in the garden or taking a walk outside, or, meditating or doing yoga or playing with your kids, or swimming in a lake. I don't know, could be so many things. I think the more and more research on immunity shows that we can prevent a lot of illnesses through our healthy lifestyle. And so it's funny, I started out studying medicinal plants thinking I was going to figure out how all this complicated, using complex plants as medicine, how all that works in a reductionist scientific way. And now I'm a little bit more inclined to say, I love doing science to try to discover molecules that might be drugs someday. I don't know if I'll ever achieve that, but in my personal life, I rely less on scientific evidence of what is good for my overall immune health and preventing of disease, and more on just simple, obvious things like eating a healthy diet and taking care of myself. So that's kind of where I've ended up with in this. Don't get so sick that your immune system is all shot and you need a silver bullet. Try to prevent that from happening by taking care of yourself. But there's also genetics there. So we don't want to blame the people who are sick for being sick. But I think, those are my thoughts on that, Ruben. Great question.
Nadja, there's a question from Maheen Umar. What was the most exciting or essential thing that you learned throughout your journey in your research?
Let me see. Okay, so what was one of the most exciting or essential things I learned throughout my journey in my research?
Yeah.
Specific to echinacea, I think the moment when we stopped thinking all of our results were coming from the plant and realized that there was microbes involved, that was mind-blowing. It was a total shift in perspective. So that was pretty exciting. I still don't know what that means in terms of how it interacts with the human, but it was really exciting to finally have an answer to such inconsistent and confusing data that we kept getting literally for years before we came to that realization. But I've had many exciting moments in science. I think often they're little things that wouldn't really mean much without giving a whole lot of context around them. But every time you discover something that is not previously known, it's a really beautiful moment to think, wow, we as humans didn't know this before, and now we do. And now I can't wait to tell everyone about it. And so that's part of the joy of being a scientist, is getting to make those new discoveries. And sometimes they're just very little things, but they sort of help us piece together what is, I think, only a little slice of our understanding of the big, beautiful, complicated universe.
Mm-hmm. Yeah. Well, I think that this has been a wonderful conversation, wonderful questions. I love the storytelling. big round of applause, whatever motion people need to make.
Thank you all.
Yeah, thank you so much, Nadja. And let me hand things back to Dan. Maybe Dan, you could tell us what our next offering for Let's Learn is. That would be fantastic. Oops, you're muted, Dano. And
Our next, there I go. Yeah. Our next free offering at Let's Learn is actually, by coincidence, has two things very much in common with this class. It has to do with plants and animals and science. It's called Science in Your Kitchen. And it's going to be led by our own Nick Gross, who is with us today. And he's going to be working with Simeon Bittman, who is a chef, and they're going to cook food. And as the food gets prepared, Nick is going to talk about what chemical reactions are happening in the food. So for any one of us who cooks, and I think many of us do, I think it'd be very interesting. And Nick, maybe you want to say a little more about it.
Yeah, so you connected me with I actually didn't know that you pronounced it Simeon. I thought it was a version of Simon, but Simon's the executive chef at a small liberal arts college in Vermont, Sterling College, and so among other things, and he's got a career in cooking, and we'll talk a little bit about that, but also as he cooks things, we'll talk about things like what's happening with any chemical reactions or biological reactions. We're looking at some physical reactions, so looking at emulsions, looking at the Maillard reaction for browning, creating those wonderful flavor molecules and what that requires, and all of that. So there'll be sort of a running commentary. We'll also talk about different ways that you can see food, right? And so science is a particular lens that we can see food through. Maybe I'll go on the defense of the reductionist approach to the scientific reductionist approach, and then I'm a big fan of that.
I'm a fan too, Nick.
Yeah, I know.
It also pays the bills.
You can't run a mass spectrometer without thinking about being a reductionist, right?
I think we can be both at the same time.
And just so you
Reductionist and holist.
yes. And that class is next Saturday, a week from today, April 1st, and it runs from 11 a.m. to 12:30 p.m. Eastern US time. And I can't refrain from pointing out that the class after that also has something similar with this, because it's about storytelling.
Oh, nice.
It's called A Tale to Tell, and it's with Ron Short, who is a master storyteller from Eastern Kentucky, and a lot of his traditional stories that he tells have become the basis of plays that were created by the Roadside Theater in Eastern Kentucky. And so he's going to share some stories and some music on his banjo, and also encourage an environment where we all get to tell some stories that were passed down to us. And that's on Tuesday, April 11th. But you can find out all the details by going to our website.
Awesome.
And we'll send out a little reminder, to everybody here. Yeah.
Yeah.
Well, with that, again, thank you, Nadja, for this wonderful presentation, discussion, storytelling, scientific explanation. Really wonderful. Have a lovely day, evening, morning, wherever you might be. Take care of yourselves. We'll see you next time. Yeah. Bye-bye.
Hi, everyone. Thank you again for spending time.
Thank you.